For the printer, it's not ART, assisted reproduction technology. Yeah, okay. Assisted reproduction technology. So, what is assisted reproduction technology? What do you think assisted reproduction technology is? Reproduction, like using technology for reproduction. Now, there are special techniques which are used. The first one is test tube baby. First one is test tube baby. Why it is called test tube baby? Is the baby inside the test tube? Is the baby growing inside the test tube? Yeah, for a few days. Few days means for embryo or for the zygote. First, the fertilization is done inside a test tube. In a test tube, they will do fertilization where the sperm and the egg will fuse, and it will be either in zygote stage, they will transfer it into a female, or a embryo stage, like 8 to 16 cells. Then they will transfer. Then we have gamete intra-fallopian transfer. Gamete intra-fallopian transfer. So the gamete is transferred in the fallopian tube. Okay, gamete is transferred into fallopian tube. Now these are all full form. GIFT, gift. Okay. Then intracytoplasmic sperm injection, ICSI. Okay. Intracytoplasmic sperm injection, ICSI. Artificial insemination technique, AIT. These are all full form. AIT, artificial insemination test. Now see this picture. This, this lady is Lucy Brown. This is the first test tube baby, not this one. Huh? This one who is almost 35 above. This lady is Lucy Brown, which was the first test tube baby in 1917 and something like that. Okay. And she is already giving birth, means she is reproductive healthy now. That was the first test tube baby. Name is Lucy Brown. Now IVF. What is full form of IVF? In vitro fertilization. In vitro means in test tube. What is test tube made up of? Test tube is made of glass. So in vitro means inside test tube or in glass. You know what is vitrified? Where this word is mostly used, vitrified. Tiles, they are called as vitrified. That means it has some shiny, glassy, shiny. So vitrified. So in vitro means in glass. Here, please write in vitro on top. You can write somewhere in vitro. Here, right this side. Right, in vitro and in vivo. Right here, in vitro. In vitro. In vitro and in vivo. INVITRO is in vitro, and there is something called as in vivo. In vitro means inside glass. In vivo is giving natural condition. Now the baby which develops in the womb is in vivo. A plant which is growing in garden is in vivo. A plant which is growing in tissue culture is in vitro. Some plants are grown in tissue culture inside a test tube. That is called in vitro. A plant which grows in garden is called as in vivo. Let's see now what happens. Now male gametes and female gametes, what they will do? They will take out. Don't write the points, huh? PPT you take. Male gametes and female gametes, they will remove from the couples. Then what they will do? Generally what happens, male gametes and female gametes, they fertilize to form zygote. But in this case, they will remove the female gamete, they will remove the male gamete, they will allow it to fertilize outside in a test tube. They will develop the zygote for 1 day, a few hours, and then they will transfer it into the female womb or female reproductive system. These are the sperm. This is ovum, and this is injection where the sperms are injected inside the ovum. The sperm is injected inside the ovum, and then this structure is allowed to grow in this liquid medium. Okay, or it is allowed to mate. Sperm is removed, and then it is allowed to mate, and it is kept in. All the conditions which are given here are exactly as the conditions which are inside the female womb. The temperature, the pressure, okay, the osmotic concentration, all will be equal to that of the womb. Then this will be transferred into a mother. What is that mother called? Sometimes a female cannot give birth, or sometimes the mother. Then we use separate mother, which is called as surrogate mother, surrogation or surrogacy. Why test tube baby? Now, why there is need of test tube baby? First, there is no conception. The female cannot conceive. Okay? The female cannot conceive. Now, there can be many reasons for female not to conceive. First reason is blocked oviduct. The oviduct might get blocked. Second, there will be spermicidal secretions in vagina. The spermicidal secretions which are there in the vagina are killing the sperm. They are not allowing the sperm to come on top. Or third is low sperm count. In males, there can be low sperm count. The sperms are not, the number of sperms are not enough. So in this case, what they can do, they can take out and do the fertilization. They will do the fertilization. Low conception, blocked oviducts, and spermicidal secretion and low sperm count. So they will take out the sperm from the male, they will concentrate it by centrifugation or different techniques, and then they will transfer it into the female, or they can use in vitro. They can fertilize it outside. The sperm and ovum, they can fertilize it outside, or they can inject the sperm by artificial insemination. They can inject the sperm by artificial insemination. Okay. Now, sperm and egg, zygote will be formed. Zygote intra-fallopian transfer. So there is one called as zip, where the sperm they take out, the egg they will take out, and they will fertilize. The zygote will be produced, and this is called as zygote intra-fallopian transfer. The zygote will be transferred to the fallopian tube. Okay, they cannot transfer it to the ovum. Sorry, to the womb or to the uterus. They will transfer it to the fallopian tube. From the fallopian tube, it will come down. Okay, then it will trickle down, and then it will get implanted. Sometimes they transfer embryo. Okay, sometimes what they will do, they will allow the zygote to develop to 8 to 16 cell stage, 8 to 16 cell stage, and they will transfer the embryo. In this case, it is called as intrauterine transfer. If zygote is transferred, it is called as ZIFT, zygote intra-fallopian transfer. And if the embryo is transferred, then it is called as IUT, intrauterine transfer. So these are all full forms. This is the same picture. If the zygote is fertilized, sperm, ovum, they fertilize, and then they will transfer it. Now, success rate of transfer this test tube baby. What is the success rate of test tube baby? It is always less than 20%. Okay, very less than 20% success rate. Drugs to increase ovulation. Now, what they will do? In order to do test tube baby, there should be production of ova or the eggs. And eggs, mostly they are produced after 28 days. So what they will do, they will give drugs to the female, and they will monitor it every day. Okay? Means they will see. Now it is given, drug on one day is given. So they will call on 10th day or 11th day. They will call the female. They will do. They will use sonography. Okay, whether the machine is put inside the vagina, it is taken towards the ovary inside. It is taken towards the ovary, and they take the pictures. They see the developing follicles, whether it is primary stage, secondary follicle, tertiary follicle, then Graafian follicle, or ovulation is about to happen, they will see it. Okay? And once ovulation is done, they will take the ovum. Okay? Sometimes instead of producing one ovum, there can be many ovum produced, which is called as superovulation. Okay? Usually it is only one ovum. But because of this drug, there can be 2 or 3 more ovums can be produced or more eggs. Yeah, it's a good thing if you go for IVF. So they can fertilize 2 or 3 and they can transfer it. Now, drugs to increase ovulation. Then they will remove the eggs and keep it. Additional embryos are transferred in surrogate method. Now, what usually happens in IVF, mostly twins are produced. They transfer 2 eggs or 2 zygotes. If one is not able to do, the other one will do. Most of the time, both will do. And there are twins produced. They try 2 eggs. If there are additional embryos, they are transferred in surrogate mother. Now, this is from USA. They don't want to kill embryo. No embryo should be killed. Okay, that is their rule. So what happens? The extra embryos will be transported into surrogate mother. Or don't make extra embryo. It is just like killing an organism or killing a human being. That is what they are saying. Embryos can be frozen for later use. The embryos can be frozen and kept and can be used for later. Then surrogate mother. Sometimes what happens, the mother is not reproductive healthy enough to carry the child. In this case, the couples will look for a mother, for a female who is ready to accept the zygote in the uterus or in the fallopian tube. Then this mother will take care of the developing baby and then give the baby to the parents. Okay. Here you can see other mother. It is like other mother. Surrogate mother means other mother. These 2 couples and this is the surrogate mother. Okay. These 2 couples and this is the surrogate mother. Now there are some problems related to test tube baby. What is the problem? Raises ethical and legal problem as right over the child. Raises ethical and legal problems as right over the child, especially in case of surrogate mother. Who is the actual mother? That surrogate mother might fight for the child afterwards. Then she will say, it is my baby. Artificial insemination donor. Vicky donor, you all know the movie. In that the sperm were given from some other person. Okay? The sperm is given from not from the father, but from other person. It is fused and then transferred into the mother. Now, obviously, that sometimes that male can come and say it's my sperm, so it is my child. Artificial insemination donor. That is the 2 problem. But yeah, but what happens now? Before this, any mother who is trying to become a surrogate mother, she should sign a form saying that there is no legal rights over the child. The child belongs to the parent. Legally, they will do. Now then, gamete intrafallopian transfer. Gamete intrafallopian transfer. What happens in this case of mother? They do not produce ovum. They cannot produce ovum, but they are healthy enough to carry the child. Okay. See, females cannot produce ovum, but can give an environment for the developing baby. So in this case, ovum from outside female, other females are taken and transferred in the fallopian tube. At the same time, sperms are also transferred and fertilization will take. Ovum from donor female is removed and transferred over the fallopian tube.
Surrogate mother ovum is used?
No, surrogate mother body is only. Ovum is from mother, sperm is from father, fusion, zygote or embryo, and then transfer. She is only taking care. She is only providing the environment for the baby to develop. So gamete intrafallopian transfer. This is GIFT. The sperm and eggs are placed in the fallopian tube. The sperm along with the eggs are placed in the fallopian tube. Then we have intracytoplasmic sperm injection. What will happen? The sperm is injected in the ovum over nutrient broth. The sperm is taken out, injected into the ovum over the nutrient broth. Now you must be wondering, this is what they are doing. This is nutrient broth. The white is the sperm, it's like the zygote. This sperm and all, they are done. Okay, now what is nutrient broth? So in microbiology, you can grow bacteria either on a solid media or a liquid media. The solid media is called as nutrient agar. So in microbiology, we can grow bacteria either in solid media or liquid media. In this, there is only difference is in this they will add gelling agent. In this, there is no gelling agent. So here they will grow bacteria as well as in this they grow bacteria. If it is solidified media, it is called as nutrient agar. If it is liquid form, it is called as nutrient broth. That is the only difference. That is nutrient broth. So what in that previous ICSI, they will inseminate, okay, they will inject the sperm into the womb and do the growing in this nutrient broth. Then AIT, artificial insemination technique. AIT. Sometimes what happens, male are unable to inseminate it or there will be low sperm count. The males, they are unable to inseminate the female or there may be low sperm count. In that case, intrauterine insemination is done. In that case, intrauterine insemination means the sperms are taken, they can be centrifuged, they can be made concentrated. Unwanted liquids can be removed and directly this can be injected towards the fallopian tube. The sperms. They will take out the sperm, they will concentrate the sperm because sperm means it will have also other hormones, seminal vesicles, bulbourethral gland, that chemicals, cowpers gland, that chemicals or the secretions are there that can be removed. They can concentrate the sperm and transfer into the fallopian tube. So this is artificial insemination. Okay, this is the last slide for today. So what is left is ICS. Intracytoplasmic sperm injection.
मैम सेल में कुछ चेंज है ये